factors that determine. Okay, so we are going to continue our lesson from sex linkage, and the primary sex characteristics are related to the gamete, as we all know, and that determines the maleness or the femaleness of an individual presenting in the zygote. So that is a bit quite different from that of secondary, how secondary sex determination is really observed. Now, you know that primary sex determination, as we have said already, is determined by certain genes that are present in the zygote. But for the secondary sex determination of characteristics, they are determined by the physical features, or better still, they are influenced primarily by these characters or the genes that are present in the individual, and that call for the expression of some facial looks, some body features. For example, in the females, you have widening of hips, development of breasts, and then you also have some genitalia differences. In the males, we have the penis, the testis, the scrotum, and then that of the females, you have the vagina, the development of the uterus, and all that. Of course, they are features that are representative for, the physical expression of those genes that were present during primary sex determination. And it's observed in the secondary characteristics, the pubic hair development and all that. All right, so you have the secondary sex characters. They are related to those ones that I have actually mentioned. And the genes that are found in several alternative forms, we can just say that they are called allelomorphs, or in essence, alleles. So the alternative form of a gene that is present in an organism, we say it is the allele. So in the case of females, you have the XX alleles, and then the males, you have the XY alleles. All right. So because of the females, they are having the same form of gene, and the males, you having different form of alleles that are determining their sex. Okay, so in the males, we can say they are heterogametic. Heterogametic in the sense that they have different chromosomes. They are not the same. So you have X chromosome and you have Y chromosome determining their sex. So we say heterogametic. And that of the females, we say they are homogametic. Okay, so naturally, as chromosomes occur in pairs, alleles are present at the same locus on each of the chromosomes of the homologous pair. Now, with the arrangements that give rise to either homozygous or heterozygous condition, according to the nature of alleles that are present, we can identify these alleles on both X and Y chromosome. But when you check the Y chromosome and then the X chromosome, you have the length of the X chromosome being longer than that of the Y chromosome, as in the case of the boys or in the case of the males. So you wouldn't get all the alleles that are present on the X chromosome to be present on the Y chromosome. Let me take that again. So you have X chromosome, which is longer as compared to that of the Y chromosome. Now you have alleles at various loci on the S chromosome, and you have that on the Y chromosome. But when you check the length of the X chromosome, it's longer than that of the Y chromosome. So there are certain alleles that are present on the X chromosome that are not present on the Y chromosome. Right. And that can lead to certain characteristics being present in males, and they are not actually present in female. Not that they are not representative, but then you realize that there are alleles that will mask the expression in the females. For example, if you are considering two alleles coding for a particular characteristics, like in the case of color blindness, if you have an allele that is coding for, I mean, color differentiation or the normal sites, and then you have allele that is defect and it's coding for color blindness, you realize that with females, they could have a normal site color, I mean, being dominant over that of the defective one, which is colorblind. So the individual will be heterozygous to the condition and so will not express the character with the females. But because the males, we inherit X from the male, the mother, and then the Y from the father, you have the defect gene in case the male inherited that defect gene on the X chromosome from the mother. The individual male will not have alternative form, that is the allele, the alternative allele on the Y chromosome to overshadow the expression, or better still technically, mask the expression of the defect gene on the X chromosome. And so the individual has no choice than to express the defect gene or the phenotype of the inherited defect gene on the X chromosome. So that is why we say that the X-linked characters are more prevalent in boys or in males than in females. So the genetic material on the homologous segments, though, have fair chance to be exchanged through the cell division, but the genes are located on this part of X and Y chromosomes seem to have varying little significance. And so they are called partially linked genes. Such traits are sometimes transmitted as autosomal traits. Since they are not present on the X and Y chromosome, we refer to them as autosomal traits. But if they are linked to the X or Y chromosome, we refer to them as sex-linked. But it is more prevalent on the X, so we use the word X-linked, X-linked characters. On the other hand, the non-homologous segments of X and Y chromosomes are of great significance because through sex-linked genes are located on them, just as I have indicated. So we can use this diagram to represent how crossing over occurs during meiosis. Now with meiosis, you realize that stage of that now and rejoin, indicating exchange of certain genetic material. So if you check on your screen, you have A, you have B, and you have C in the, and the other is striped. Now if you go to the B segment, you have the unstriped one exchanging genetic material with the striped one. And at the end of the day, at C, you clearly see segments of the striped one or the striped chromosome exchanged with that of the striped one. So you have at the end of the segment of the striped one, breaking and rejoining the striped one, and then the unstriped one breaking and rejoining the striped one. So you have the striped one joining the unstriped, and portion of the striped joining the unstriped as well. Okay. So completely, Y-linked genes are also called the hollandric. If you have certain genes that are present on the Y chromosome and not present on the X chromosome, such gene is called hollandric. The genes on the non-homologous segments of the X chromosome are the most common type of sex-linked genes, and they are referred to as the X-linked traits. Okay, so I think I have explained that there are certain genes that are present on the X and are not present on the Y, and they are referred to as the X-linked character or X-linked genes or X-linked traits. They are particular and really peculiar alleles that are being posited in one of the X-determined chromosome, and they really determine these characteristics that are observed and they are prevalent in males than in females. Now let's look at characteristics feature of sex-linked traits. So you have one as the gene governing the sex-linked traits. They are not transmitted from male parents directly to their male progeny. And this is it. A father is always transmitting Y chromosome to the boy or a male child. So even if the father is having a trait or a defect gene on his X chromosome, he is not going to transmit directly to the male child because he is going to transmit Y chromosome of which is not having the defect gene. So the father will rather transmit X chromosome which is having a defect gene to the girl child. And now the girl child, when grown and also married, can transmit that defect X chromosome to the son. So it's like the father will not transmit directly to the male child or the male progeny, but it is rather his daughter who will transmit that defect gene on the X chromosome, which was given by the father, to the grandson. So it is not directly transmitted from the father to the son, which is the X-linked characteristic, but rather it is the female daughter, is the female progeny of the father who will transmit that defect gene on the X chromosome to the grandson of the father. So that is how it works. So number two, a male transmits his sex-linked gene to all his daughters. Since female receive one of their two X chromosomes from the father, like I have explained earlier, the male has no choice than to give the X chromosome, which is having the defect X-linked gene, to the daughter. But the female, since they have XX chromosome or the two X chromosome, making them homogametic, they can transfer probably one with non-defect X gene to the daughter. But a man has no choice than to transmit the one X chromosome to the daughter. So the daughter will inherit the defect allele or the defect gene born on the X chromosome and eventually transmit it to the grandson. The third one is, since X-linked chromosomes are not located in the Y chromosome, the heterogametic sex is hemizygous for each gene. When we say hemizygous, it means you are carrying or you bear only one of the allele instead of getting the pair to control a character. You have only one of the allele that is controlling a character. And that is observed in males and not in females, because the X chromosome is longer than the Y chromosome, and there are alleles on the X chromosome which is not present on the Y chromosome. Let's look at causes of sex linkage. And when we talk about linkage, we are talking about a phenomenon where alleles present on a certain chromosomes are inherited together. So all the X-linked characteristics like baldness, like hemophilia, also you have the G6PD, and then the likes are inherited together on the X chromosome to the son because they are hemizygous. And so when you inherit this defect gene on the X chromosome, you are going to really depict it or express it. So one, the location of a gene in the X chromosome. We will soon look at heterochromatin and euchromatin. Now, in heterochromatin, the genes are tightly packed, and that will not allow expression and subsequently transmission of genes on the heterochromatin are really linked up, and that it means if there is a transmission, they are going to be linked. Crossing over rarely occurs in that region. So all the genes that are present there are most likely to be inherited together, unlike on the euchromatin. Euchromatin, the maps or the map gaps or the distance between the genes are widely apart, and that crossing over is most likely to occur in that region. And the alleles are independently going to assault into different gametes. So the location of a gene in the X chromosome plays a role as far as linkage is concerned, or inheritance of X-linked characteristics is concerned. Then number two, you have absence of gene alleles in Y chromosome, like we have been mentioning. Since you don't have the pair or the alternative form of a gene from X chromosome to the Y chromosome, there are certain alleles that are present on the X and not present on the Y, and the individual is most likely going to express the presence of these alleles. All right, we can look at genetic mapping. So the sex linkage done, let's consider gene mapping. The relative distances between the positions of gene on the chromosome. We refer to them as the gene maps, or also the chromosome map. So linkage is incomplete because the homologous chromosome, when paired, can undergo an exchange of gametes during crossing over in meiosis 1. And particularly, we are referring to packaging stage in prophase 1 of meiosis. So if there are, assortments, you are not going to have sex characteristics, or better, so certain traits that are linked in a chromosome. You may inherit, but there are characteristics that are independently assorted into different gametes. And so, yes, homologous chromosomes, when paired, they can undergo crossing over, like exchange of certain genetic materials that can result to linkage not really occurring. Right. Linkage of genes can be presented in a form of genetic map, and which is also known as linkage map or chromosome map. So the relative distances between these genes or alleles present on the chromosome is regarded as the map. So genetic mapping is the first step in identification of isolation of new genes and determination of its sequences. So the distances allows the isolation of certain genes, and we can study how the nucleotide sequences occurs in the DNA. It is equally essential in human genetics for identification of genes associated with hereditary diseases. And if you know the sequences of certain character, you're able to determine at what point a change occurred that caused the defect, or that caused the character defect or variation in certain characters. And that could be detected and corrected. The concept of gene mapping was first developed by Morgan student and Alfred Stamvart in 1913. So crossing over is manifested physically as chiasma resulting from breaking and rejoining of chromatids. So exchange of these genetic portions of chromosome, as we have discussed earlier, occurs in packaging stage of prophase. The theory of crossing over is that each chiasma results in new association of genetic markers. Now, genetic markers are the specific genes with known sequences. The markers are those specific genes with known sequences, and their location is stable on the chromosome, and it is known. So when there is no crossing over, the alleles present in each homologous chromosome remain in the same combination. So realize that if there is no crossing over, these genes that are present on the chromosome are inherited together. But if this crossing over occurs, there is dissociation of portion of this chromosome and reassociation with non-homologous chromatin. So it will break and rejoin others, and you have variation occurring in such individuals. That is why you are born of the same mother, the same father, but you have variation, you have differences between you and your sibship or the sibling. You aren't the same because of this phenomena of crossing over. The portion of the chromosome that breaks and rejoin, changing the genetic makeup of individuals. So let's look at how really this occurs. Of course, we have observed in cell division. We can reconsider it on our screens now. So we have A portion and we have B portions. Now we have the A. There is no crossing over occurring in the A column, and there is crossing over occurring in the B column. So you have the blue and you have the red. Now you have the blue to be homologous and you have the red to be homologous. And you have the blue and red chromatids or the chromosome to be non-homologous. Let me repeat that again. So you have red and blue chromosomes and they are paired up, so we say homologous pair. And then we have the red also paired up, and so we call them homologous pair. But the red and the blue are non-homologous pair. Right. If you consider that of the A column, there is no crossing over occurring, breaking and addition or breaking and rejoining of segment of the non-homologous chromosomes occurring. But if you come and check the B column, you have the blue chromosomes, the homologous chromosomes, certain segments of the blue portion breaking and rejoining the non-homologous chromosome of the red. And so at the end of the day, if you check the downside of the column of for B, you realize that the new genetic material is established. You have red and blue portion on the same chromosome, which means there are sequences on the red chromosome that are found now in blue chromosome, and there are sequences in the blue chromosome that are found now in red chromosome for the non-homologous chromatids. Now, finally, if you have the cell dividing, you have an individual having portion of red and blue to the far right. If they have the cell dividing, you have an individual having both blue and red portion of the chromosome present in a gamete. So that when said, we say there is a variation that has occurred because the individual are having sequences from different chromosomes, and that is crossing over. So there are representation for genetic distances. You can have one as frequency of recombination, and you can have value like 0.024. You also have percentages of recombination, and that's instead of 0.024, you can have it as 2.4. 2.4. And you also have the third one as map distance in map units. And you have it as 2.4 map units. So we are just using hypothetical figures. So you could have it as frequency of recombination as 0.05 something, 0.03 something, 0.01 something. I mean, any hypothetical figure. But when it comes to the percentage recombination, you have it at, if you have, for example, 0.050 in frequency of recombination, in percentage recombination, you have it 5.0. And in map distance units, you have it at 5.0 map units. On your screen, you have it at 2.4 map units. And the last one that we can also consider is the distance measured in centimorgans, which is represented as small c, capital M. Okay. So, for example, in your test, if I give the frequency of recombination as 0.060 as the frequency of recombination for the genetic distance, and I ask you to convert it to map distance in centimorgan, the answer expected is going to be 6.0 centimorgans. Right. So crossing over takes place in first meiotic phase after chromosomes have duplicated. And I have said that already, in packaging stage of prophase 1. It is physical exchange between chromatids like we have seen in pair of homologous chromosomes. It is also consists of breaking and rejoining of two in of crossing over. Probability of crossing over increases as the genetic distance also increases. And this is the principle of gene mapping. So with gene mapping, it really correlates with the frequency of crossing over. Now, the wider the distance between the genes that are present on the chromosome, the more frequent is likely that crossing over will occur. And the closer the distance, the most unlikely a crossing over is bound to occur. And they are going to be inherited together. And that we say they are X-linked. They are X-linked. Or they are sex-linked. Because you can have situations where you have alleles present on Y chromosome that are inherited together. And we call them holandric, as we have. Right, so we have genetic and physical distance. The greater the physical distance, like I said, between the genes along chromosome, the greater the map distance between them. And we have discussed the map distance and their representations in map units, in frequency of recombination, and in measuring in centimorgans. So physical and genetic distance are usually correlated because a greater distance between genetic markers afford greater chance for crossing over to take place. And I think that is widely explained. Genetic markers, I said, these are genes that their sequences are known and their location on the chromosome is also known and it's quite stable. So if you check the distances, if the distances between them are quite wider apart, you have crossing over most likely to occur. But if they are closer, you have crossing over most unlikely to occur. So frequency of recombination between genes may differ even in sexes, because they can have different map distances in their genetic maps, although the physical chromosomes are same for both. All right. So let's consider a chromosome. So you have chromosome on your, and portion of the chromosome, we have euchromatin and you have heterochromatin. In the mid portion, and you have euchromatin in the other extreme. So if you consider these two portions, you have the darker and you have the red portion. The red portion is labeled as the heterochromatin. Now the heterochromatin portion, the map distances between the genes are very close. And transcription is most unlikely to occur. That is, copying of segments of these genes or the segments of this chromosome are even most unlikely to occur because of the map distances. Very, very, very close. But when you consider that of the euchromatin, the map distances between the genes are wider apart. So in this region of the chromosome, crossing over is most likely to occur. So with heterochromatin, the distances are very close, and chromosome or chromosomal crossing over or segments crossing over at this point is most unlikely to occur. But it is frequent in the area of euchromatin because the map distances are wider between the genes. So gene mapping in human pedigree. So genes, one, the genes that cause human diseases are rare and are observed in only small number of families. Two, many genes of interest in human genetics are recessive and are not detected in heterozygous gametes. And you know that in every character or characters are controlled by pair of alleles or pair of alternative form of genes. In heterozygous gamete or organisms, you have a dominant allele and you have recessive allele. And then dominant will always mark the expression of the recessive allele. So you, even though the individual has inherited the defect allele, but the individual is just going to be a carrier and not going to express that defect character. Three, the number of offspring per human family is relatively small, and that segregation cannot be detected in shapes. It may be very small, minimal. The distance may be very minimal. And then four, human genetics cannot perform test cross and back crosses. When we say test cross, we mean that if somebody is exhibiting certain traits or a dominant trait, phenotypically, you would want to ensure and be sure that either the person is homozygous or heterozygous. And for you to do that, you use one of the offspring who is exhibiting a double recessive inherited alleles. So for example, let me just use practical example. If you have tall parents, you are not sure whether the individual or the parent is homozygous tall or heterozygous tall. That is the individual carrying two tallness alleles or one tall, one short allele. So you would want a child who is exhibiting shortness. If you exhibit shortness, which means you have double recessive allele, short and short. So you cross the individual with the parent and the offspring. If any of them exhibits shortness phenotypically, it means the individual who is exhibiting or the parent who is exhibiting tallness or who is expressing tallness phenotypically is not really homozygous but is heterozygous. That is test cross. It is not done in humans. You cannot do that. Also, back cross, an individual exhibiting a desired characteristics, the one of the offspring is used to cross the parents to maintain such characters in the family. Of course, if you have a German Shepherd and you cross it with a local dog or local breed, and you want the characters to maintain in the family after their first filial generation of after the offspring, you use one of the offspring to cross the parents. So if you use one of the offspring between these two hybrids and cross the parents, you said you have done back cross. So we do back cross. We embark on back cross to maintain desirable characteristics of a parent. And it is not actually also done with humans. You cannot use any of the offspring to cross the parents. It is not allowed. So recombinant DNA technique allow direct access to the DNA. Genetic mapping in human pedigree has been carried out primarily by using genetic markers. So we don't use or we don't cross individuals just because you want to study them, but you can extract their genes, study their genes, study the sequences of their genes, and of course, you can do genetic recombination rather than crossing two individuals entirely, as in the case of human, to assess their pedigree. There are many minor differences in DNA sequences from one person to the next, very minor. So the difference is not much. We check the genome and the sequence that exists in our genome. The differences are not really much. And it is approximately 1 in every 1,000 base pairs. That is where we have difference. So you can have in 1,000 base pair sequences, you can only have one difference. You can only have one nucleotide difference or change in only one of the nucleotide sequence, which is out of 1,000 sequences. You can have only one change, which means these changes are quite infinitesimal, even though you are considering it to be quite infinitesimal, but the impact is also huge, as observed in the people with sickle cell anemia. You have the change in the glutamic acid or the substitution of glutamic acid for valine, and that has ever caused their woe. So as we end today's session, this morning's session, I would want us to go through the definitions. Crossing over, we have discussed that. Cytokinesis, we have discussed that during the cell division. Look at enzyme genotype. Look at gene mitosis. We've considered that. Meiosis, we have considered that. Sex chromosomes, I think we have considered that. Phenotype, somatic cell, and hybrid. So those ones that we have considered already, we're just going to highlight them and discuss those ones that we haven't really come across them. So crossing over, of course, exchange of genetic materials between non-homologous chromosomes. That can be determined or called as crossing over. Cytokinesis, the division of the cytoplasm of a dividing cell, or the splitting of the cytoplasm of dividing cell, can be called cytokinesis. The division of the nucleus is called karyokinesis, and I'm sure you have that in mind. All right, so enzymes. Enzymes are biological catalysts that speeds up rate of reaction occurring in organisms. And we have many of these enzymes, both plants and animals. And in plants, you can have hexokinase and the rest. In humans, in digestion, during digestion, you can have amylase and the rest. Genotype, you have the genetic constitution of the organism. In the genotype, you have two pair of allele. Oh, sorry, the pair of allele. When I say two pair of allele, I mean four, but I'm referring to only two alleles. So a pair of allele. If it is tallness, you have, and it is homozygous, you have it as capital T, capital T. But if it is heterozygous, you have capital T, small t. But you cannot walk to any hospital and say, I'm coming to check my genotype. That is so wrong. You can only say, I'm going to check genotype of my blood group, genotype of my height, genotype. So when you say genotype, the character that you want to check the genotype of must follow. Genotype of my sickling status. So you cannot just walk to the hospital or health facility and say, I'm coming to check my genotype. That's the mistake most people do. It's not their fault to do. So gene, we are talking about a portion of a chromosome or a portion of a DNA that code for specific characteristics that are inherited from parents to offspring, or that are transmitted from parent to offspring. So certain specific portion of the DNA that code for specific characteristics that are transmitted from parents to offspring. We regard it as a gene. So mitosis is a type of cell division that produces two identical daughter cells. In the end, the chromosome number is maintained. You have 46 of the offspring or in a daughter cell, and the parent is also having 46. So we normally refer to it as equational division. That of meiosis is reductional division, because at the end of the day, 4 daughter cells are going to be produced. And the chromosome number, since it is reduced, you have also crossing over occurring, and the daughter cells are genetically non-identical, because since there was crossing over, there are portions of the chromosome that will break and rejoin, changing the genetic sequence. And so they refer to them as non-identical. But 4 daughter cells are going to be produced. You have sex chromosomes, and these are chromosomes that determine the sex of an individual. And in humans, you have Y and then we have the X chromosomes. We've spoken about that already. Phenotype, we are referring to the physical expression of the genotype, the genetic constitution that I have made mention of. Its physical expression in an individual, we refer to it as phenotype. Example, if you have a genotype of OA, which means your blood group is going to be A. If you have AA2, your blood group is going to still be A, because A is dominant over O. O allele is recessive to A. So if your genotype or the genotype of your blood group is AO, you are going to be A. If your blood genotype is OB, you are going to be B. But if you have B and A or AB alleles inherited, you are going to be AB blood group, because A and B, they are co-dominant alleles. They all express themselves equally. And so you're going to be AB blood type. You have somatic cells. These are cells that do not determine the sex of an individual. So you have body cells that are somatic cells. So somatic cells are referred to as body cells. They don't determine the sex of an individual. The one that determines sex of an individual is referred to as sex cells. And for example, we have the sperm and the egg to be sex cell. Some of the somatic cells, you have the epithelial cells, epidermal cell, epithelial cells, you have blood cells and all that. These are all somatic cells. So you have monohybrid cross. This type of cross, you are considering only one character. This type of cross, you are considering only one character. For example, if you are crossing two individuals, because you cannot cross human, you can only do genetic recombination, like we have stated. You cannot intentionally cross two people for a study. I mean, humans for a study. You can only do genetic recombination. But in plants, you can afford that, or we can afford you that to cross two plants. And so if you're crossing two plants to study only the hybrid of flower color, you're only considering the flower color, white and let's say red. You want to cross a species with red flower and another species with white flower to study the outcome of the offspring or the phenotypic expression of the offspring. That is referred to as monohybrid cross. You're considering only one character, that's the flower color. But if you are considering, the height of the plant and also the flower color or the pod shape and then the seed color, the same cross. Or if you're using the cross to determine the pod shape and the pod size, you're doing dihybrid cross, or better so you're embarking on dihybrid cross. We call that cross dihybrid cross. And so people will end a 1 hour session here. This evening, we'll continue the session from where we left off this morning. And please, I'm coming. I've seen your hand. And finish up. So I will give you some assignment to do even between now and the time that we meet at 7. So heterochromatin, I have explained that already, but I'm just asking you to explain. Euchromatin, linkage map, homologous recombination, and then also X-link inheritance. Kindly do that. These terms, I just want you to explain these terms that I have given you and submit them to my mail. So you have the mail. Kindly, even when you are sending, kindly pay attention to detail. If you are sending the mail, kindly, or if you are sending the web, kindly pay attention to detail. You have D dots. I'll call you, please. The one whose hand is up, I'll call you. You have D dots, bats dash, Kobina at stu dots, ucc dots, edu dot gh. Please pay attention to details when sending the mail, because if you input wrong email address, it will not come. It will not come. So please pay attention when you are sending it. Thank you very much. All right, I've seen a hand. Can I have the hand again?
Yes, sir. Sir, it is
Okay.
me. I want you to repeat the part that you have corrected. That we go to the hospital to check the genotype of our blood, and then you mentioned some other ones, but I've forgotten, and like it's not clear for me. So please, can you repeat that part so that I will make that correction if I also visit the hospital or whatever.
Right. So what I'm saying is that your characters are governed by genotypes. Your characters, every character that is constituting you is governed by genotype. Your hair color, your eye color, your blood type, your sibling status, everything, your height, okay? Everything is governed by genotype. And the genotype, we have alleles that controls a character. Kindly mute yourself. So you have a character that is controlled by two alleles, that is a pair of allele. One can be recessive and the other one will be dominant. Or you may have two recessive alleles, or you may have two dominant alleles controlling a character. And that is your genotype. So, for example, fair skin and a dark skin. When you go to the hospital, you are going to check the genotype of your skin color. Of course, if you are fair, why do you go to hospital to go and check the genotype of your skin color whilst you know you are fair? So most of the times you go to hospital to check those ones that are not really visible to us. For example, your blood type. You don't know your blood type, and it is not written on you when you are going. Nobody sees this is your blood type or your blood group. So you go to the hospital and ask them to determine the genotype of your blood group. Okay. Now, you also go to hospital for them to determine the genotype of your sickling status. If you are AA, it means you are normal. You are no even a carrier. If you are AS, your genotype is AS. It means you are a carrier to the condition, sickling condition, but you are not expressing it, or you will not express it because you have the dominant allele masking the expression of the S, which is the recessive allele. And if you have SS, it means you have double recessive allele, and that you have the condition, or you have the sickle cell condition. And so that's what I'm saying. You go to the hospital for them to check your genotype of a particular character that you don't know. Please, do you understand what I'm saying? You don't walk to the hospital and say, I'm checking my genotype. Because the genotype is your genetic constitution of a character. Please, do you understand what I'm saying?
Please.
Yes, sir.
Okay. Please, do you understand what I'm saying?
Yes, please.
Yes, sir.
Okay. Any other question? Okay. If there is no question, kindly go through this and define them, explain them, and then you can send them to me. Check the details of the mail again, the email I have sent, and then kindly send your work there. Thank you very much. If you face any difficulty, to kindly communicate to your course rep, he will call and then we can discuss it. We'll meet at 7, 7 to 9 p.m. today again. Thank you very much. This class was for last week. Last week we lost 1 hour, and so we were supposed to make up for the 1 hour. And so that is what we have done. Thank you very much.